Thank you. I'm super happy to have been invited to that session. My English clearly doesn't qualify me for that, first. Second, it's end of the afternoon. You might be tired. I try to be quick, and those who know me already know I'm lying. Anyway, so I didn't choose the title of my topic. I think it's Isabelle. So it was a challenge for me. It's nice. So observational data for precision health. When we don't know exactly what to say, we start by defining things. Raphaël said it for good reasons, like we might not have understood what she says, or the same reproducibility or replicability thing. And for me, it could even give me the possibility to end my talk now. Like, precision health aims at tailoring preventive and therapeutic interventions to each patient rather than using the same approach for everyone. And this would use genetic, lifestyle, behavioral, environmental factors. So it broadens the scope of precision medicine to what occurs also outside the settings of the doctor's office or the hospital. So we may consider also disease prevention, health promotion activities. And it's not complex to argue that we will need observational data to build and evaluate precision health, just for the reasons that have been said before. Like, if it's difficult to do trials for a very specific drug in an apparently broad population, it will be super complex to do multiple randomized controlled trials assessing part of interventions, targeting some features to build the whole of the precision health. And if we agree now, I can stop. Then you save 20 minutes. Or we can go a bit further. It's difficult for me to talk a lot of precision health because I have not started working on that. But what we have been done in the team for a few years now, it's working on precision medicine. So let's take first a step back to precision medicine. It's precision medicine or personalized medicine? I don't want to debate with Babak or others. Like, just the European Commission says personalized medicine, and they define it as an integrated package of healthcare solutions comprising elements of medicines and medical devices that are structured to meet an individual patient's need. That's quite specific. Take a simpler view: to use individual characteristics to base medical decision in an organized manner. Otherwise, it's just medicine. And there are many situations. You may say, I will base precision medicine on the knowledge of the pathophysiology and the mechanism of my drug. This is more molecularly targeted medicine. But you could say also, I will do clusters of patients with either similar characteristics or a close predicted response to the treatment. Then some said it's just doing smaller strata. That's stratified medicine. But if you use complex models, it's also precision medicine, and then you could even go to tailor interventions and adapt treatments to the individuals, and that would be more personalized treatments or individualized treatments than individualized medicine overall. Often, if you search Google for personalized medicine, you will find delivering the right drug to the right patient at the right time. But that's not so easy. Do you think you have a drug? No, you have a patient, sorry, and you want to find the right drug for that patient. And this has led to umbrella trials, for instance, in cancer, or observational data use. Or do you have a drug, and you want to find the right patients for that drug? But if you are a drug company, it's just what you want to do. And this has led to basket trials in cancer, but also more classical randomized trials, observational data too. Okay. Let's go a bit narrower. Take the easy. I'm not claiming that doing molecularly targeted drugs is easy by itself, but the situation is easy. You know more or less the molecular target, the biomarker, that I will call BM, because I'm lazy. And then, what type of designs would we use? Well, we could use a biomarker certified design. You test the biomarker for everyone, and then you randomize people, possibly stratify on the biomarker value, and you randomize them between the experimental and control, and this will allow you to test the interaction between the biomarker status and the treatment effect, like whether the treatment effect is the same for patients who have the molecular target and those who do not. That would be super useful. Or you could even do biomarker strategy designs where you test the biomarker, and instead of randomizing everyone on everything, you randomize patients to either a marker-based strategy, the personalized medicine strategy, you only give the new targeted molecule to the biomarker positive and the other gets the control, or control for everyone. Or you have another version of this one where you randomize people between testing the biomarker and then going to the biomarker strategy, or not testing the biomarker. And those designs where they allow to test the effect of adopting a personalized medicine strategy, and they were the preferred one in the PERMIT European H2020 project where I was leading the clinical trials part. And especially our HTA colleagues in the project were super in favor of these ones. One thing is that for that we may like those biomarker strategy designs, they might be super inefficient. If you have a rare molecular alteration, the difference between the experimental and the control arm is only for a small epsilon% of your sample. So overall, you're comparing two groups who basically receive the same treatment except for a small portion of them. So it would be nice for HTA to have such trials, but clearly it's difficult to fund because nobody does that. It's super rare. And if you look further, even the evidence behind the biomarker-based approvals in the US is low also. And this has been done by a former PhD student of the team who has worked on that a long time. For many studies with molecular targeted agents, what occurs in practice is that people use enrichment designs. They only randomize biomarker positive patients. And often they do not even randomize, but Sylvie told us before that this occurred. And this is also true for drug licensing in cancer. So if we want to find reasons for that, because the molecular alterations are rare, because sometimes there are high response rates in population with unmet need, and sometimes because, well, I think Sylvie has given some clues, but actually this is what occurs. And there is here a first place for using observational data to complement or to supplement the unobserved control group that we would have liked to have in a randomized trial. So in the single-arm trials, you have no counterfactual, so you can try to use external data, why not observational data, to find the control that is lacking. This is not easy to do. There are many methodological and statistical challenges, but at least methods exist, and I can refer you to a paper by Jérôme here. Well, but the truth that if we want to go for precision medicine, it is not only always one biomarker. I said many things at the beginning, like behavioral, why not genetic, but also beliefs, other characteristics. So it's much more complex than just having one biomarker positive, one biomarker negative. And then you cannot do biomarker stratified designs anymore, because you have many potential biomarkers. So there have been many statistical machine learning methods that have been proposed to build models to predict the drug efficacy based on the individual characteristics of individuals that I call X. You have really many paradigms, algorithms. I'll be reassured I will not enter into that, but you can try to estimate what would be a good rule to assign to match the treatment to the patient characteristics, or just estimate what would be the treatment effect given those characteristics, and then a natural choice would be to to select the one which gives the best treatment outcome. And to start from the easiest, we have tried to do that with many algorithms using RCT data. So we got rid of confounding. And if we do that, and here we have taken the 2 large randomized controlled trials, you will see 22 algorithms in row and columns, and the green or bluish colors, they give you a hint of how how concordant the resulting individualized treatment rules were. And what we see is that it's the same for the 2 trials that have nothing to do. One is a very old stroke trial, the other one is a traumatic brain injury, more recent trial. Is that you have big batches of algorithms, that the algorithms that have been ordered by closeness, so the ones who are just small versions of each other, they agree very well with each other, but the others, they completely disagree. That's not the data on which the models were fitted. Flory, she cut the data into 2 pieces. She trained the algorithms, could be very simple statistical models, more complex machine learning on a part of the data, and she tested what would occur in the remaining part. So this poor agreement was puzzling, and we say, well, are we doing nonsense? Or perhaps we are trying to look to very, very small effects, and one trial is not sufficient, even if it has a 20,000 patient sample size. So we went to IPD meta-analysis, the meta-analysis on individual patient data. We've talked about that, you've heard about that a few minutes ago with Philippe. So we didn't try to get the data, that's a huge endeavor. We just benefited from someone who did that before. What's nice is that we have more data. We also have more variability, which might be nice to increase the external validity of the models. And all were randomized trials, so we still do not have to bother with confounding. What's nice also is that you can do something like internal, external cross-validation, like you fit model in all trials -1, and you test in the remaining one, and you do that many times. Of course, you have to account for the added complexity of accounting for heterogeneity in the models and avoiding aggregation bias. But what's nice is that Rachel showed you models of heterogeneity, so we went for the tau square, but you can do that for many regression parameters. It's complex, but you can do that. And then we also used numerical simulations, and on the simulation, it works well, or pretty correctly, depending on the algorithm, the sample size, and so on, but it's okay. But when we got to real data, well, things were super messy, and clearly we could not find a model and find an individual treatment rule. So perhaps we need for, again, larger amounts of data, and this comes to a second option for observational data to derive individual treatment rules. You've seen Josh. He was able to have access to 136 million patients. Of course, they do not all have traumatic brain injury, but perhaps it will be more than 9,000, and perhaps the increased sample size would make it possible to fit those models to those data. But so technically, or at least statistically, it's possible. Like all the algorithms I've shown before either are straightforward to observational data or need to be adapted for that, but this exists. But now you will have, of course, to account for confounding, and it's not so easy, we've seen before, but at least you could try to do that. And then you will have all the methodological consideration to account for like emulating a target trial. But again, this is possible, at least if we trust or we are willing to trust. But that might be done. Just a couple of hints. In many statistical papers, they just use the bunch of covariates as X. But when you want to tailor interventions. You may have some variables you want to tailor your intervention on, and some others you want to adjust for confounding, but you would not want to tailor the intervention. And for instance, you might have big gaps in the distribution of socioeconomic status between treated and untreated patients. You have to account for that, but do you think you would recommend the nice drug for the rich and the other one for the poor? I'm not sure, so I leave you with that consideration. Up to now, I just forgot the red. Right drug to right patients, not easy, but then we have at the right time. Wow. But of course, many treatment decisions are not decided at a unique time point. And in Josh's example, you have new users, right? It's new users, initiators. This means that those guys, all the months before, were non-initiators. They were controls somehow. And then once you have tailored initiation, you have to tailor stopping treatments, unless you want to treat people for life. And this leads to what is called dynamic treatment regimes. Like you have to decide when to start the treatment, when to stop it, and for whom. You have a specific randomized controlled trial designed for that. It's called sequential multiple assignment randomized trial, SMARTs. Not the SMART of CRESS, another SMART. But they are super rarely used in practice, and a recent review showed that, quite paradoxically, only 17% of those trials actually aimed at identifying the best dynamic treatment regime. Well, that's puzzling for me, because this is what the design aims at, but that's the matter of fact. So if nobody does trials, you can either do a SMART trial, but think twice, because even the people who have advocated the design do not do so many SMARTs, or you will have to resort to observational data to derive your DTR. And there is a whole range of methods, not much about from biostatistics field, but more from econometry, but or game theory, but you have many approaches, and one would be reinforcement learning that I will present you. So I will switch to an example of a former colleague from the team who tried to build a dynamic decision tool for initiation of renal replacement therapy in the ICU, like for adult patients with a stage 3 acute kidney injury in the ICU. And it took 3 days. And he assumed that after 3 days, he was an ICU physician by training. After 3 days, let's assume that either they should have had RRT or they will not need it anymore. And every day you have a decision to take whether to initiate or not renal replacement therapy for them, and we want to know for whom we should initiate that. He used a large electronic medical records database, which is called MIMIC3, which is super used in practice because it's available. And it took as a outcome the hospital 3 days at day 60. And then the objective was to find a dynamic strategy that would maximize the average HFD 60 for the whole. And for that, we used what is called offline Q-learning. I'm sorry for the words. It's not so complex, actually. Like, you have to find an optimal sequence of decisions, and that's not so easy. But then you start by the last one, day 3. And here, you just have a single point decision. You just want to identify who would not be under RRT there. Among those who are not under RRT, who should start that day? And then you fit relatively simple regression models for that. And then you assume you get the optimal choice. Then you go back to day 2, and at day 2, you do the same, but conditioning on the fact that you assume you will take the best decision at day 3. And then you go back to day 1, and if it was more than 3 days, you would do that. You start from the first one, and you always condition to the optimum in the future to optimize the current one. I'm almost done. And then well, you can always do that. Like you will get a model. It's super complex to know what it does. So we used data from the control arms of two other trials to compute the mean difference in HFD 60 if you followed the strategy that was built like that, or versus the control group of the trial, which was found considered as a good standard of care because it's a control group of a nice trial. And then we also compared with another strategy, which would be start everyone at day one. There were two strategies. I was lying a bit before. Like either you decide to take the treatment which gives you the best a mean of HFD 60, or you add confidence intervals and you want to only start if you have 95 confidence that you should do that, which is called the stringent policy. And if you do that, or the first one, well, okay, if you look at the confidence intervals, we did not formally show that the personalized strategy was better, but still, at least it's clearly not worse, and you save RRT for some patients if you do the stringent policy, like you only treat half patients, and probably possibly you do better. Interestingly, a colleague, I would even say a friend, Stijn van Stelen in Ghent, he was involved in that work, which was very, very similar. They wanted to find a dynamic treatment rule for initiating RRT in patients with stage 2 and 3, slightly different population. Acute kidney injury in the ICU, and they did something completely different. They said, we want a stationary rule, like every day you have the same rule of whom should start RRT. In the previous study, it could change from one day to the other. Perhaps on the first day you would be more stringent, and day 2. Here you have the same rule, and they did that figure, the decision tree, where everything relies on yes or no conditions and 4 thresholds for pH and potassium. And they combined all the possible thresholds for those 4 values for the 4 thresholds, and they ended up in emulating 81 emulated trials that would each use a different combination of the thresholds. And if you do that, you can compute the average mortality. I think it was ICU mortality in that study. You can compute the average for all 81 trials, and you might take the lowest one. So, emulating a target trial of precision medicine is possible. This was the approach chosen by our Ghent colleagues. You can do that in a static setting, like you have a one-point decision, or in a dynamic setting. And then you could replace or supplement the lack of trials evaluating those strategies. So while perhaps we may wish that the final evaluation of a DTR would be based on randomized evidence, before going to that, you can test a lot with observational data and at least not choose some that clearly show it would be a waste of resources to add that arm to a trial. The clone sensor weight approach, and I hope you slightly talked about not much. I hoped you would do more, and I would have to do less. The clone sensor weight approach is very well suited to DTRs too. Like you clone patients, you create clones. It's super nice because you have no ethical concerns at that step. You have many others, but at that step, no. You clone, you put the clones in each of your arm, even 81 that works, and then you keep the clones in the arm until they deviate from the strategy, and at that point, you censor them, and you have, of course, to account for the selection bias that you induce by this censoring. There are also ways to evaluate ITRs from observational data in a more complex framework. And I will refer here to a paper with our colleagues that it's a bit complex to read, very complex to read, but super comprehensive and defines different estimates according to what you consider as implementing a rule and what is the control. Just be careful, even in the ones before, the same individual patient can be in different arms in those emulated trials. Because if you say start renal replacement therapy if pH is above 7, and someone started with a pH above 7.3, he or she will comply with that rule, but will also start with pH more than 7.1, 7.2, blah, blah, blah. So the same individual could be kept in the different treatment groups. As a conclusion, I hope I was not too long, there can be other uses of observational data for precision health. You can project or transport the treatment effect in an other population. I think we already alluded to that slightly before. We've done that with Philippe in hypertension, and Alex will do that within the More Europa project. I would try to advocate that observational data are key for precision health, at least for the development of tailored information. There is no other way you will get the data. You need large sample sizes, you need big streams of data coming from wearable sensors, omic, microbiota, whatever you want. This will raise methodological challenges in how to measure that, how to operationalize that, how to use those methods. And you will have to harness a huge complexity in terms of variety of the intervention, variety of the tailoring variables. But clearly, we will not have experimental studies for that. Then we come to the evaluation of tailored interventions, and here you have undoubtedly a room for RCTs. First, of course, you have RCTs for new drugs, new devices, but then you might also have RCTs to evaluate a personalized strategy, but you need first to construct it. Even at that stage, observational data can play a role, as they already do. And I will finish there. Thanks for your attention. And I just thank all the colleagues who work on that